Hello, everybody. My name is Ethan Moyer. This is, it's October 6th, 2021, the fall term of Drexel AI. And it's week 3. In this meeting, we'll be talking about, more progress toward the project. Vivek and I kind of individually figured out what exactly we'll be looking at in regard to the data set, and we'll explain for our reasons why I'm doing that. And I think that we all have some things to talk about. I think Alex has a few things he wants to mention, and then Vivek has some things, and I have some things. And I hope, within this week, I can really, Vivek and I will focus on pre-processing the imaging data. I might be able to open that up on my computer. I just haven't been able to do that lately. Vivek, what's up?
Hey, what's up?
I like your lights.
We were just doing that. That's why I got late.
Yeah, that's okay. It's lit, dude. Your room's already more stylish than mine.
Yeah. And I have Abby with me.
Hello.
Cool.
In the meeting.
Cool. We can start whenever. Do you want to go first, or should I go first? I already did introductions.
You did.
Yeah.
So, hi everyone. I'm Satvik. I am the treasurer at Drexel AI, and we are okay. Hi, Brandon. So we would be working on the basic architecture of the project, and we would be setting up a repository on GitHub so that we could start coding. And I would template for the research paper so that we could start adding stuff. Yeah.
Is that okay? So that paper template, is that on your
Yeah.
overleaf?
I'll share.
Can you share that with me? And I can share my screen. I'm gonna have everything open here. Assam, what's up?
Oh, hello. Hello.
This is my email. So yeah, we were just talking about how we want to get started this week on processing the data. The clinical data and those features are easy. You're really only going to deal with the numeric and categorical features, and you can easily encode those into a meaningful vector. The genomic data is easy. The genomic data is really just a matrix. I can go into this right now. Yeah, so the genomic data, it's called RNA-seq. The way it works is you have, you basically look at what RNA transcripts are expressed in the genome, and you do this through a method called short read sequencing. So you cut up the genome, you cut up the RNA reads that are being expressed, and you read these short reads. The reason why you read short reads instead of long reads is because as you read longer reads or longer RNA sequences, the quality begins to greatly diminish. It's very difficult to actually read long read sequences of DNA. You can align these reads according to a reference genome, and then what you'll find is you'll see that, okay, so in this region, there might be a lot of overlap with this gene. So you'll see like a high peak, and then maybe there's not so much in this area of the genome. So you'll see a low peak. There might be a lot of overlap here in this gene. So you see a high peak. And then what you get is a matrix. You're going to do this for many genes, about 22,000 genes over many samples. And each sample here corresponds to a different individual. And you get a matrix corresponding to the read count. I think it's like RPM reads per million. So like the columns are samples, samples, and then the rows are just each gene. So this might correspond to one gene. This is another gene, and then this matrix is what we can use. So these are all just numeric with some missing values. So it's a pretty straightforward data set to use for AI. The one data set that's not useful for AI right now. Oh, who's that? Andy.
Hello.
Sure.
I didn't realize I was not muted.
No worries. You realize now. Cool. Anyway, the one data set that isn't directly prepared for AI or takes the longest to prepare, in my opinion, is the images. I don't have them up right now, but we can, I was looking at some, these are MRI. Our images look pretty similar to, I want to find it's pretty similar to these right here. What's up, Adam? Oh, sorry. Pretty similar to these DICOM images. DICOM is a standard way of representing images. It's used for the DICOM standard controls where metadata is and how the pixel array is represented in data. I found this tutorial that talks about using some things from SK image for image segmentation. I think that Savek actually gave us a really good figure in the group chat. Sorry, my computer's been very slow and sharing my screen.
Ethan, did you try figuring out the segmented images? Did you try plotting them? Like how?
I didn't plot them. I'm just assuming that the images are already segmented and we can just use them outright.
I wanted to see how's the segmentation.
Okay, I can look at those this week. I just want to make sure that everyone's like on the same page with what we're doing, because we're dealing with like 3 major modalities here. But you know, we have the features for the clinical data, easy to encode those. We have the features for the genomic data, easy to encode those. This is kind of our wish list for the imaging features, right? You have the easy ones, which are just volume, surface area. There are some other ones that are more difficult to generate. I don't actually know what these are yet, but these are supposed to quantify different things about the brain, like different things about the lungs, like coarseness, contrast, busyness. I don't know how you quantify busyness, complexity. So, I mean, over the course of the next full few weeks, we'll work on developing code to, you know, extract these features from the images. Yeah, any questions about that so far?
I have one question from me. So is there a data set with the, sorry, I haven't been here, but is there a data set with like the lung images with the RNA data and stuff?
Yeah.
Oh, okay.
Yeah. So right here, it's 75% of the images are segmented. It comes with like clinical data. This clinical data isn't like our clinical data, Samu. It's very low res. It's like whether or not they're a smoker. It's like their age, stuff like that. And then 130 of the subjects have RNA-seq data. And that's that matrix I was talking about earlier.
Okay. And for the RNA part, you talked about like cutting it into like small, or like, I guess you kind of like highlight different parts of the image of the RNA, right? Like given like a sequence.
That's how RNA sequencing works. So let me see if I can find a good image that displays it. No. I don't like any of these images.
Overall, is it like finding the frequency of each type for each like section?
Yeah, it looks at the amount of reads.
Okay.
This is good. So yeah, what you have is you have these RNA transcripts, and you can dissect them, and then you can sequence those RNA reads. And when you overlap them to some, you know, when you overlap them to some reference genome, you can say, okay, you know, there are so many reads that overlap with this region. That means that this region is highly expressed in this tissue. And then you like look at the RPM, which is the reads per like amount of reads per million. That's the workflow.
Okay. And basically kind of like the RNA is separated into these kind of like different types, kind of.
Yeah, because the RNAs correspond to positions along the genome,
Okay.
and you can use something called like sequence alignment. There are a lot of sequence alignment algorithms that attempt to align that sequence back to the genome.
Oh wait, align the sequence. Okay,
Yeah,
so
because you have
like
to figure out from what genome each read came from.
okay, so it's kind of like, so each of those lines are like different gene, kind of like genomes.
Different. I don't know if I'm explaining this correctly.
I just don't know much about biology. TBA.
Yeah. So there are different sequence alignment algorithms that allow you to align those RNA sequences, because you have these RNA transcripts, right, that are just, sitting in space. So this one's just in space, and using the sequence of the RNA transcript, you can tell me where it is on the genome, like what gene it came from, because you transcribe genes to RNA, and that RNA is then used to encode proteins. We're intercepting the RNA in the cell, asking from where it came. We're asking, where did this RNA come from in the genome, because those are the RNAs that were expressed. And then we can say, okay, it's from this gene, and if there are a lot of RNAs expressed at this gene location, then that means that gene is highly expressed, and we can show that in the matrix right here.
Okay, and where does the alignment come in?
After the short reads have been sequenced, after the RNAs have been sequenced.
Okay.
Yeah, cool. Any other questions? Cool. Savek, yeah, he put in a. Wonder if can I add this somehow to mine? Is there a way to do that? I have no idea. Sorry?
I think it would get added as you will edit it. Like
Okay. Okay, cool. So this is a template we're going to use. It just says default values, but we're going to just use the template because it's easy. So a couple things. This is Overleaf. Overleaf is a online editor for LaTeX. This is an archive template. Archive is where we publish all of our work. When you publish to archive, it can then be accessed on Google Scholar. So that's the goal. And I have the conference proceedings still on hold for some reason. I have to come back to that. I don't know why I archived. I don't know. I don't know how we were supposed to do the conference proceedings. I'm not going to waste too much time on it. We'll just come back next year. So anyway, I wanted to just get a GitHub started for everybody.
Also, Ethan, I thought about what should be the title, and it should be like risk prediction for a
Yeah.
And I was reading through an article, and it said so far people have been using the metadata such as age, sex, and smoking history. And nodule size, morphology, and growth from CT, but nobody has used the radiogenomics features.
Yeah.
That would be really cool if you use that for the risk prediction.
It would be.
So we could just use the people who are alive as the standard and the people who died as the predicting value.
I was talking to Alex about that. I don't like those endpoints. I really don't like those endpoints at all, because if you have an 85, if you put surgery, if you do surgery on someone who's 80 years old and they die 5 years later, you have some sort of edge case like that, then you don't know if that was because of the cancer or because of just old age,
It shouldn't be too difficult to work in some heuristic to account for that, though, right?
What would be your heuristic?
Maybe weigh them less severely the older the person is.
I don't know, man. It's it gets difficult. I don't want to give the model. I mean, should the endpoint be death, or should the endpoint be something closer, like whether the cancer will reoccur?
That does seem like a better marker.
You don't want to just create a we don't want the model to learn death, right? I don't think that's the goal of the project. It'd be kind of dreary if we just would create a model and it'd tell you when a patient's going to die. It'd be nice though if given that they had cancer, we learn whether that cancer is going to reoccur, and if it's going to reoccur, when is it going to reoccur?
We could also, depending on the type of cancer, account for the stage, how
Yeah,
big
so
the lesion or the
it's called grading. You can do some sort of grading like that. Age limit. You could use an age limit, Andy, but it gets difficult because people can die from unknown causes, and I don't think in this data set we have the cause of death, right? I mean, if you crash your car the day after your surgery, you didn't die from the surgery. You died from a car crash.
Are you sure?
Yeah, just that's that. So I have the data downloaded. I think I mentioned this to everybody last week. It's on a external drive right here. Drexel AI, radiogenomics, single cell data. We can actually look at the data. I'll show you. This is what it looks like. This is what the that's a bad representation. This is what the genomics data looks like. It's a matrix. R1-23 is a patient, and this is a column corresponding to a patient. Each row is a gene, and then these genes have different strengths. So you might ask, what is the maximum value in the first column? That would be the gene that was most highly expressed in that patient. You could and then you could easily encode these features. I mean, you have missing values, but those missing values most of the time are for every
Is it just 0?
So here's the interesting thing, Osamu. So the way that a lot of biological data works they end up so this data has already been processed once, and what they do is they do peak calling. So you might have, if this is the genome on the bottom, you might have some zeros or some noise, but then you might have some peaks and you might go back to 0. So what they do is they call these peaks at the genes, they report the heights, and then they map everything else to NA.
Okay.
I'm pretty sure that's how they do it. So you can still use the NA values. Oh no, we hear some background noise. Who's the culprit? I'm coming for you. Don't Hong. Thank you. Sorry. Yeah.
Okay, but
so
for the purposes of this thing, you just set it to 0, I guess.
We can, I think.
And also, how did you download that? Like, I'm looking on the site, and the only thing there is search for RNA.
The RNA is actually hosted on a different website. It's hosted through this thing called Geo.
Okay.
How did I get it? Actually, where is it? Oh, right here. Yeah, it's hosted on Geo.
Yeah. Okay.
The Geo is a publicly available consortia through NCBI for data for published studies.
Oh, I found it now. Yeah. Thank you.
My PI at my lab at the Children's Hospital actually downloaded all of the methylation data available on Geo, and we have like 161,000 samples to work with, which is pretty cool. Not samples, studies, I think. Yeah, something like that. So it goes over the methods, what they used. I wonder if they did any pre-processing on it. They did something with statistic. They did like a t statistic. So maybe we should do something similar. You encode all of the the features into a t statistic, and it seemed to work for them. So we'll probably do the same thing instead of using like absolute values, but still that conversion is relatively simple, right? That's where I downloaded it. Anyway, so that is the genomic data. I think I did this last time. We can look at sorry, I was reading the chat. We can look at the clinical data too. There's two files. I think this is the clinical data. One CSV file is controlling the metadata for the images, and the other one's controlling the metadata for yeah, this is the metadata for each patient. So you see we have the person, where they were located, how old they were, their weight, male or female, ethnicity, whether they were a smoker or non-smoker, and some other things like type of cancer, some genotype information, tumor location. I think there was a grade. This is a bad way to view the data. I'm going to open it up in numbers.
Open it on a Sheets or Google.
Yeah.
Sheets.
I was.
I have
Yeah,
one.
you're.
You need it? Wait a second.
That's okay. It'll be open in a second. And this data is relatively easy, Alex. This is the one I'm talking about. Like you can just make this categorical, and that's easy to do. You just gotta be mindful of the NA values here. And we'd be trying to predict this right here. This would be this is what we use for our endpoint, the date of reoccurrence. We don't have that for all of that. That's kind of the downside. So maybe we could look at, time of death and then age and then date of reoccurrence. You could even use survival status as that too. So you get more samples out of survival status.
Why don't you just look at recurrence?
Oh, because you get NA sometimes. Oh, NA means that it doesn't reoccur.
No, recurrence like 2 left.
Oh, this one works as well. I'm blind apparently.
Yeah, and it does it for almost everything, I think.
Yeah.
Like
well, one thing, if you only look at recurrence, right, is you're treating all yeses as the same, which is difficult because there's obviously something different about a cancer if it recurs in a month compared to 10 years. There's something different about the biology.
Okay, then maybe how about both recurrence and date of recurrence?
Yeah, probably.
Yeah.
So that data is easy to encode. For anyone who actually wants to look at this, like Alex, if you want to look at these features, do correlations and code them, I would just try to get as many of the columns as possible that are not unique. For instance, like this one's unique and this one's just like perfectly non-unique. Actually, sorry, this one is actually, you could use this as a feature because it's just 2 sites. If you need help, I can do it, or give you pointers. But you may have done this stuff before. Yeah, and then the other thing that is interesting is we have metadata for all of the. Sorry, this is to download the data. This is the clinical data. This is genomics data. This is the manifest. No. We have different files for each patient, and it's gonna be too difficult for me to view it here, so I'm just gonna use the online viewer. It'll be much of the same anyway. I'm just scrambling around. I think somebody posted a link to the data set. Thank you, Alex. Yeah, so there's this search button here. I was using like py.com, which is a nice viewer in Python that allows you to view the DICOM images. We should be able to look at some here.
You could just use the DICOM viewer on here.
Yeah.
It's easier.
What is?
Search results. Here. Yes.
Sorry.
Search results right next to the summary.
Oh, okay. Thanks. PT. So this one. CT Fusion. Oh, sorry. Yeah, so we've looked at this before, but this viewer is very useful. You can change contrast. You can
And-
evolve.
You could even segment it and add like region of interest on it.
Oh, from here,
Yeah.
there are some pretty easy ways to segment just using filtering, which I might just do instead.
Like Segmentation, we could get some manual segmentations as well.
Yeah. So like my worry is that just takes a long time to do.
Really?
But I mean, you have to get a clinician. I mean, would you happen to have a clinician? Do you? But if this would load, I'd be able to show you the lung. And I was reading before this meeting actually about how they segment MRIs and how they actually segment the CTs. They mentioned how depending on the region, there's different windows that you're supposed to look at depending on what you're looking at. So like fat is around this area, the lung is around the -500, so that's where we would look. The lung, and then they use they use like those thresholds to segment the area, and even to segment the lung itself by using a function from sklearn called find contours. So I'm sure we'll just use the same thing because it seems to work pretty well. I mean, this is the result. It can even find these 3 areas when the lung is cut off. Like you can see it's snipped right here, so it's 3 instead of 2, like you'd expect it to be. You can get a region of interest like this, and then you can even look at the blood vessels too here. And you can do different kind of thresholding so that you only get the blood vessels on the left instead of the edges of the lung on the right. And then this is what it looks like when you put it back onto the lung itself. So I'm sure we're gonna just use this. I mean, I think this is a good first step than just trying to jump in and try to go all out with this without we want to make sure we understand it first, and get some experience with this kind of technology. So yeah, right here is the lung. It's a lot of images, so it's gonna take a long time to load, but yeah. So I created the GitHub, right? We have a GitHub created now. I cloned it, and what I want to do is I want to come in here and I want to just dot up some things. So we should talk about tasks this week. Okay, so we found the data set. That's good. I will just put a link to the data set here. We have the data set. We have a paper template. I'll put that here for everybody who wants to view. I'm just putting, I know this is obvious, I'm just putting it in one central location. Could you send me the public link, Vivek? We need to analyze or sorry, pre-process clinical features. We'll worry about correlations after we have all the features. Pre-process genomic features, pre-process imaging features. This is honestly going to be the bulk of the work. It's what's going to take the longest time. I can give this to people if they're interested in it. Alex, do you want to name to one of these? Would you be interested?
I'll take on the clinical features.
And I can do the imaging features with Sadek if anybody wants to try it out. I mean, I have good resources for what you could use. This one is probably the best, at least for the CT scans.
I could also probably help out if we needed to, because I've done like stuff with filters, OpenCV, so that if that's any of help.
Okay. Osamu, have you done any image processing?
Yeah, but I'm taking 19 credits this term, so
Okay.
I'm not sure.
Yeah, we'll
Yeah,
we'll
sorry.
leave you alone then.
Yeah.
If anybody else is interested, like, speak up. We can schedule another 30-minute meeting at the end of the week or something to talk about and to actually execute it. I don't think it'll take that much time to do. We just need to figure out how we're going to calculate all of these features, or as many features, or a number of features that make it meaningful in the data set. So then after we have all these features, we're going to process or just calculate correlation, and exclude highly correlative features.
With medical projects, like having good visualizations of how the features are looking in the data set, that is really important. Like how are they scattered in relations to one another?
Yeah, I was gonna show some of them in like a couple minutes, but having it for the rest of the features is really important. I agree.
Yeah. So you mean like clustering?
Yeah,
You can do like PCA or something.
Like distributions, yeah.
there are actually more. Forgetting the names.
Hierarchical clustering, distributions. That is good. After that, after we do that, we calculate correlation, exclude highly correlated features, do clustering. Sorry, I'm just gonna add this so it makes sense. Then what we can do is we can just throw machine learning algorithms at it, so we can fit a model. Since we're dealing with a highly diverse set of features, I'm going to just suggest a neural network. We can look at decision trees, or we'll just do random forest. Better regression.
Yeah, I had a question about this. Like, would you want just, I would imagine you wouldn't just want one model for everything. You'd want like ensembling different things, like for the clinical features and for the images, or like two separate models.
I don't know, actually.
I guess it just depends on the correlations that we find, as
Yeah.
well as how you do with the images and the DICOMs.
I've never done a model like that before.
Actually,
I wouldn't know how to set one up like that, actually.
Yeah.
I've always done something end to end. I haven't segmented it.
I have worked with like such model before, but we hard coded it. So
Yeah, so it's a lot more difficult. Yeah, so these are the main tasks. When we get to writing the paper, that one is a little more
production.
background.
Can we do
but
two
the-
separate, like introduction and background as two different
You
sections?
want to do that?
Yeah, because I was reading through the guidelines and Elsevier needs that. Methods.
Then we need an abstract. And then we need figures.
Do we need any tables?
I mean, that'll come up with, this is the bulk of the work. So like, did you send me the link to the,
Yeah.
can you send me the link to, is that the public link?
It's in, yeah,
Okay, thanks.
I've also sent a link to the clinical data. I uploaded
Okay.
it to Google Sheets, so it's in the chat.
Oh, silly me. Where is,
It's on Zoom.
yeah, I don't think I can copy it. Let's try it again. I feel like an old man learning how to use Zoom. Okay, there we go. There we go. Okay, cool. Yeah, this is the bulk of the work. It's week 3. There's many of us. I hope we're all excited. I think this project is going to be really good. For people who don't know, the reason why we picked this topic is because, I mean, Savek found it, and I like when data sets are available, but this was released on June 1st. And data like this we don't get data like this too often. It's 3 different modalities with a relatively clear endpoint. I say clear endpoint because in the real world, you rarely get good endpoints in medicine. Surprisingly, Osamu and I, we work with traumatic brain injury data. We have, a data set of 124 patients, but 13 of them have a well-defined endpoint. So you can't do much machine learning with that. There's other ways to measure, there's other ways to do prediction with that data, but not as interesting.
I think Moberg like sponsor Drexel AI now.
Sorry?
Moberg should start sponsoring Drexel AI.
You have no idea what's going to happen in the future. So just you wait.
Really?
Oh no. Don't worry about it.
Okay.
So these are the tasks. Oh, that's silly. That's silly. Okay, I know what happened. These are the tasks. And we can, get started this week. I told this Savek, I'm submitting a grant to the Department of Defense at the end of tomorrow. It's due tomorrow, so I have to finish. So after that, I'll have a lot more time to work on this, and then we will. For those who are interested, I can. That obviously did not work. What am I missing? Was I supposed to do something else, or are there just spaces after these or something? Or maybe it's not after these. No, I really messed something up here. That's so silly.
I think the dash goes before the brackets.
Sorry.
The dash goes before the brackets. So it'd be like a hyphen kind of, and then a link to our bracket, X, totally to our bracket. Does that make sense?
Oh, I see.
Yeah, like that.
And I still need I still need the slashes at the end, I think.
Weird thing.
Oh yeah, it's marked down. Yeah, so I'll do this. Alex, do you want to share your screen and talk about it?
Yep, I'll go ahead. So I'm just going to show you all what I found for the clinical data. There we go. We all love Markdown. So yeah, I just downloaded the CSV file onto this Colab notebook. And the cool thing about this is that I can share this notebook with all of you. You don't have to download anything. It's just, you just open up on your drive pretty much. So you can start working with it. But we got all these columns here, and we got about 39 usable features that are non-unique. And patients are either from Stanford or I think VA probably means Virginia is my guess.
Veteran
so-
Affairs, maybe.
Oh, Veteran Affairs, that's true.
Or actually, no, if they're not associated with the military, I don't think it would matter. I don't know.
Oh, we didn't care.
My guess probably Virginia. Yeah, it could be either one of those, but who knows? This is what Ethan and Satvik were talking about earlier with like just predicting death. And it's that most of the patients we're dealing with are about like 60 to 80 is like majority of them. So there could be a lot of other causes, a lot of other health related causes, or just not health related, just general causes that kill them. So that's why we should also focus on recurrence. We could still work with the death date, but we should also focus on recurrence and other things as well.
Hi, sorry, one question here. Who's that one person at 0 years old?
Oh, no. It's someone who's 24 to 25.
Oh, 24, okay.
It's just the way this is. It's just because it's just the way the access is. But yeah, most of these patients.
Okay, I
most of the
thought it was
patients-
like a infant smoker or something.
I sure hope not. But yeah, 24 to 25. Most of them are kind of in the older after 50.
On their way to dying,
And then most of our weight, this 0 is just missing values. Don't worry about that. Nobody weighs 0 pounds, but also the weight distribution is fairly like normal. I would say we only have a couple like overweight patients, but that that probably might affect the lungs. Like, I would imagine it would, but who knows? We'll see based on the
It definitely
correlation.
does.
Yeah, because you probably have a lot more like fat that just reduces. You'd probably better explain it than I can, but yeah.
You should, yeah, you have fat pressing up against organs, which is, or pressing up against the lungs, and you can't inhale as much oxygen and you have to breathe twice as fast. Something else. So was there height in this?
Was there height? I
Yeah.
don't think so. I can
That
check.
stinks, because if there was height, what we could do is you could do BMI. The reason you want to include BMI and height and weight is because most models can only learn linear representations of features, and BMI is a non-linear feature composed of height and weight.
Yeah, unfortunately, I don't think we have height, but
Thanks.
yeah. Besides weight, ethnicity and gender are here. Specifically, I remember last time we talked about smoking status, which is something I'm definitely going to look into, because obviously if someone's smoking, their lungs are probably not going to be as good, and they're probably going to be more prone to lung cancer. And based on our data set, they're mostly former smokers, and there's also some non-smokers and current smokers. That's just that graph right there. Years patients have been smoking for. This is pack years. That's my guess what that means. I'm not 100% sure what this means, but
Pack years.
yeah, because it would make sense for someone to be smoking for 120 years. That doesn't, but I don't really know what this means.
How many
so.
packs they smoke a year?
It's probably like the number of packs you'd expect to smoke in a year and like times however many packs they actually had.
Yeah, maybe.
Like
It is basically the number of packs of cigarettes smoked per day, multiplied by the number of years the person has smoked. Oh,
So number per day times per year. Okay.
yeah.
Okay.
Yeah. So can you go back to the distribution of sex up there?
Of
So
gender.
male, female. Okay.
It's a bit more male than female, but yeah.
Yeah.
Like we have the ethnic classification. Can we also do something like talk a little bit about the ethics and fairness related to the model?
We
Yeah.
could, because
We might want to exclude those two features out in one test.
Yeah.
because the majority of them are Caucasians. That might be an overrepresentation.
Well, I want to see how they work.
Yeah.
Yeah. That's something we can talk about in like the conclusion of the paper.
Just to be careful, I recommend seeing like the proportion of people who have recurrence for like males versus females, or like smokers or males versus females, because I think in the RNA there might be some like parts that are like indicative of that, like male, female, right?
Hmm.
And
So-
like it might make a model that just classifies males versus females instead of smoker versus not smoker.
Yeah. Asamu, when we deal with RNA data, when we deal with genome data, we always exclude the X and Y chromosomes. So that's
Okay.
an important note.
Okay.
And no other basically no other genes encode for sex besides those.
How about the race and stuff? Like
The races are here, so mostly Caucasian, some Asian, African, but it's generally not proportional distribution, but
I'm just wondering whether like the genomes and stuff kind of like have race information in them.
So
true.
the question, I sat in on a lecture, and what this woman did, I think she was from Harvard, she actually took a genome, someone's genome, and she could tell us what regions belong to what ethnicity.
Oh, really?
Yeah. Like this ethnicity was, Caucasian, this ethnicity, like this leg on the chromosome was Filipino. It was crazy. So yes.
Like Stanford released this model which can predict like the race of a person through their X-ray scan of chest.
Okay. Then I wonder
Oh.
how you prevent that on like.
Sorry.
Yeah.
Some of that, the data that she did it on was DNA methylation, which is highly affected. It's more granular than RNA sequencing because you have
Okay.
more sites. You have about 28 million sites as opposed to
Oh, wow.
22,000 features.
Okay.
I don't think so. You'd see a lot of ethnicity effects there, but typically on the genomic level, you don't see genomic effects. You only see genomic effects at these places at variant sites or what are called single nucleotide polymorphism. So they're like DNA, they're like point mutations. You don't see that in the RNA expression because you only see the number of transcripts.
Okay, so basically kind of like if you just exclude the ethnicity part, then you won't need to worry about like a person's ethnicity in that.
Sorry?
If you exclude the ethnicity from training, you don't need to worry about ethnicity kind of reoccurring in like the genome part.
I don't think it's going to be a problem in this case.
Okay.
Because our data isn't granular enough to predict ethnicity.
Gotcha.
And with that, I think that's pretty much most of the visualization I've done before, only because the rest of the variables are categorical. And obviously there are ways to visualize categorical values. I just have to convert them to numbers with data preprocessing. And especially for the smoking status, that's especially where one hot encoding is useful. Because obviously, if you're a current smoker, that's a lot stronger of an effect than if you're a former or if you're a non-smoker. That's where you would use that in. But yeah, and the correlation, this is a correlation matrix I tried to make, but it didn't really work out because I still need to convert them into values because they're kind of weak right now. I'll get better results on that. But yeah, go ahead.
Yeah, a couple things. So yeah, missing values, categorical mapping for, you mentioned, what was the one you mentioned for like patient status? You mentioned
smoking status.
Yeah, smoking status. What did I say? Patient status? Yeah, smoking status. One kind of encoding you could do that I just thought of would be interesting for ethnicity. I mean, obviously we know that certain ethnicities are closer to each other than others. So like if you think of like, I don't know, human evolution, right? Some ethnicities are closer to other ethnicities. So it'd be cool if you could weight those ethnicities by how far they are. So like you have one ethnicity, one, and there's one ethnicity that's super close to that ethnicity. You'd weight it. The weights would be more in congruence than if you had an ethnicity that was farther away. I don't know if there's research on that or if that's possible at all, but that would be something cool to do. You would likely need to find a paper like weight bias, like weighting ethnicities by phylogeny. Also, for these numerical features, make sure that you standardize them. So just convert them into a statistic.
Yeah.
You know, just you can assume just we can just play this game. You can assume that these distributions are all normal and just, you know, do that. I know that the pack years isn't normal, but you can just, it's fine.
For our purposes, it's normal. But yeah, that's pretty much what I'm going to do next. If any of you who haven't been to the last meeting want to join in, you can. I'll probably be working with us on the Slack channel, or you can just join me or join Satvik or Ethan's group. Just let us know. Let us know which parts you want to work on, and yeah, that's pretty much what I wanted to share you.
Cool.
Yeah, I recommend also looking into imputation to kind of like fill in missing values and stuff.
That's another thing, because I know there's like you can do it just replace with mean or median. There's a bunch of other different ways, so I'll have to figure out which one to do.
Yeah, one way it's just replace with zeros, I guess. I don't know.
I guess gets pretty silly. I don't know if I'd want to do that. I don't know. I get funny with imputation. My PI at CHOP doesn't like imputation for that very reason, that you're kind of just manifesting data that isn't there. Cool. Thank you, Alex.
And for the images and in the sequencing data, I wonder how far like CNNs and stuff would you, but that's just a suggestion pretty much.
Well, Osama, we're not going to use the CNN on the image itself. The reason is that what you meant?
Yeah, like the DICOM image and stuff.
Yeah, the reason we won't do that is because the images are large, A. B, the ROIs are different image to image. So you really need summary statistics about each image to have a finite set of features. That
How different are the ROIs?
I mean, if you have a collapsed lung, that's a lot smaller than a lung that is normal.
Okay.
And I might be twice someone else's size. You might put me up next to a petite woman, and you know, my lungs are just massive and hers are just small. You know?
Yeah, I'm just wondering if like you can get some of these statistics that denote a collapsed lung. Like that's good enough without a CNN. Like I know you can get some, but
yeah. But then there's also the computational requirement of using a CNN on all those images that are all high resolution.
yeah, that's fair.
Yeah. I think for a first pass, learning how to encode these images will be a good exercise for all of us. There are resources, there's a SK image, but the fact is we're using these simple tools on a new data set, which is particularly interesting. It's not, you know, it's a set of data that I'm sure many of you haven't worked with in tandem before, all at once. Cool. Okay, so imaging, RNA sequencing, clinical data. I have to admit that I really don't like when they say clinical data. I don't like that. The clinical data that Osama and I work with are much higher resolution. We have many modalities, upwards of, you know, I don't know, 20 modalities per patient. And each of these modalities are collected at like anywhere from 1 hertz to 256 hertz if it's EEG. It's a lot more high resolution than these clinical data. So the day when we can have these high resolution clinical data with imaging and genomics, I'm so excited. But this is fine for now.
We could rather call it metadata than.
Yeah, it really is metadata, but we'll call it clinical data in the paper because that's what everyone else is used to. They're not used to the future yet. Cool. Sweet. So we have a plan. We have a data set. We have actually like the 3 types of data, draft paper. All that information is on the GitHub. I'll post the GitHub, the README on the Slack channel. Feel free to clone it, make a branch. I don't know if do I have to add you as contributors. I don't know how this works. Maybe. There we go.
Thank you for the call out.
Cool. If you have any questions, you can just stick around and I'll be here for a while. Otherwise, we can talk in GitHub, we can talk in Slack, and I'll see you next week. And for people who want to work on the images, I might schedule a call later this week as well. Yeah, thank you for hanging out.
Yeah.
See ya. So like you heading out?
Well, I think it's
Heading
going.
out?
Yeah, it's going great.
Abby, are you biology?
My major?
Yeah.
No, I'm a comp sci major.
Oh, sorry, I wasn't sure.
Do you see the dinosaur? He's a biomed major.
Who? Oh, cool. I think I met him.
Yes, you did. He's my room.
Okay, cool.
I don't know very much about computer science coming in. I just knew that I like it. From
Yeah.
what-
Sorry, Safek just said you could help with the biology, so I that's why I assumed biology.
Not she.
Oh,
My girlfriend could. She
Oh.
did join in. Oh, after.
dude, too many girls. You got me confused.
No, just one. Actually, her grandma, she's going through like lung cancer, not lung cancer.
Non-small cell.
Yeah, breast cancer. No, not
Oh.
small.
Sorry.
She had her chemo, so she actually knows a lot about cancer.
Okay. I hope her and her family are doing okay.
Yeah. So yeah, she would probably be taking charge of bio because I didn't get it either. Like I know a
What?
basic,
You know a fair amount.
not a lot. Yeah,
Cool.
basic.
All right.
Also,
Thanks for.
you could use sparse coding.
Yeah, we can use sparse coding. I don't want to get into that yet.
That would be fun.
It would be fun. I'll suggest it in a couple weeks after we get some real progress.
Is there anything I can do to help you guys as a beginner who like
Do you know Python?
I kind of know Python. I kind of hate it, but
Oh,
I really
geez.
I'm a really fast learner. I
You should see what you can learn from Sadvek's Google Colab, because what he did is a basic data analysis that like most people should be able to do.
Yeah. She could help in writing the paper because you don't need.
Yeah, you can help in writing the paper.
Yeah.
We'll be using machine learning models on the data, and we'll want to have a section where we explain the rationale behind using them and what they are and a little background. And I can help teach you guys. I'm going to teach about the models because I understand them, and I'm sure Sadvek understands them as well.
Also, you pronounce my name wrong, like all the time.
Can you tell me?
Satvik.
Sadvek?
Yeah,
How do I say it?
you say something different.
Savek?
Yeah, that's wrong.
Sadvek.
Yeah,
I'm so sorry. I'm actually the worst when it comes to pronouncing names.
you have a bad accuracy.
Yeah, bad accuracy.
Really bad.
Cool.
Yeah. I literally joined 4 labs and now I kind of realized they were saying the right thing.
Me?
Yeah, it's overwhelming.
You do pick 3 things that overlap. The first thing makes you money. The second thing is a club. The third thing also makes you money, but it's fun.
I did 2 labs, which is paid, 1 is unpaid lab. Yeah. And yeah, that's it. Plus 3 minors.
Yeah, you should be careful.
Yeah. But this Concord collaborative lab is so good. You might have heard of it.
Banu.
So they're really nice. They work with good stuff.
Cool.
Have you ever been to College of Medicine?
No, my girlfriend wants to go there for med school.
I want to go there to become my lab coat. But it's so far away.
Yeah, good luck. Cool. I'm gonna upload the video to YouTube.
I'm so hungry.
Sure.
Yes. I'll
I'll
see
see
you.
you.
Bye bye.
Bye.